again, we're going to start to basically explain how to use these genomic prediction models in the proper way in your breeding programs. But what has to be recognized and remembered is that we need to have a training population. Basically, this can be the phenotypes and the genotypes that come from your program when you are, in the stage 1, for example, of yield testing. You are always generating phenotypes. You just have to genotype those individuals, and all that information, phenotypic and genotypic information, will become your training population. We say that all that information is going to put in a model, or being put in a model that probably later we're going to touch some details, and also Abby probably will touch on that. And we're trying to predict some individuals that you will not generate probably a phenotype, or although you could, normally you don't want to invest, let's say, more resources. You're going to genotype these individuals, and you're going to use this model to predict their breeding value or their total genetic value to take any of those two decisions, select for population improvement or advanced products. If that is still on your mind, then let's move to really the first application of genomic selection. I know that was kind of a long introduction, so now let's focus on how the breeder's equation is linked to genomic prediction. Sorry. Okay, so let's focus first on the accuracy component. That's why I'm highlighting here accuracy in the breeder's equation. As we were mentioning, by the point that you are considering to include the genomic information, you have probably already taken other steps to increase accuracy in a classical fashion, let's say. You are, doing better agronomic practices. You are implementing better experimental designs. You are covering better your target population of environments. You are using selection indices. So all those things that I'm mentioning are still in the classical approach. And you expect that, you are already applying all those things by the time that you say, I already applied everything. What else can I do? Well, how can I use genomic prediction now? Then is the right time to start thinking on that, not before. So then let's put it more graphically. We're talking about this, Mickey Mouse breeding scheme. You have your crosses, you generate nurseries, and then you move through different stages of selection. And we're talking about, doing better crossing and using better surrogates of genetic value. So for example, instead of using just the phenotype records, maybe you calculate a blue, or if you are even better, you calculate a blob. And if you have even more support, you calculate a pedigree blob. In the classical approaches, you still don't need any genotype information. And if you are in the optimized field trials, maybe you are adopting P-reps or augmented designs in early generation trials and things like that. And you are reducing your generation interval, maybe by not waiting to a lot of inbreeding, or just identifying how to propagate faster your materials through clonal propagation. I know some of those methods for potatoes, jam, etc. But when we are talking about using genomic prediction just for accuracy, we're gonna focus not on the fancy stuff of genomic prediction. Normally, the first step for making sure that you adopt genomic prediction is to just use a better surrogate of genetic value. Let's assume that, like I mentioned before, you already are at the top of the classical programs. You are already using a pedigree blob model. You're not using means, you're not using blues, you're not using blobs, you're using pedigree blobs. That's where you are before you start thinking, how do I use genomic prediction for increasing the accuracy? And when you are in accuracy, we're gonna focus here. Later on tomorrow, we're gonna focus here, and we're gonna focus here. So better intensity and better cycle time. But for now, we're gonna focus on the surrogates, and I'm going to explain a little bit on selection indices, if the time allows. So basically what I'm trying to say is that you're gonna use the genomic information to generate not pedigree blobs anymore, now maybe G blobs. for the individuals, maybe in your nursery stage or in your stage 1 or in your stage 2, your stage 3. It's up to you. You can actually use them in all of them. So I will show you the results from some of the simulations that we have run for different crops, for line crops, for clonally propagated crops, and for hybrid crops. And I'm gonna try to go slow. So in this case, if you're really missing something, let me know, because this figure is going to have a lot of information. But we're gonna go step by step. What I'm showing you here are the results, like I said, we did a lot of simulations for different programs. We have many reports, but I wanted to bring a picture that I feel that summarizes a lot that you find in most of the crops. So this is a genetic model that we simulated to understand how genomic prediction helps to maximize the accuracy in breeding programs. And don't focus too much for a minute in the figures, I will explain them. We simulated a breeding program that basically during the first stage of yield testing is growing, let's say 1,000 entries. You already went through the nurseries, you already discarded everything for disease resistance, etc., architecture, and let's say you already have 1,000 materials that you want to send for yield testing. I know that this might be too much for some, too little for others. It depends on the crop. But what we are going to see here is the use of 3 different surrogates of genetic merit for the traits of interest, which is blob, pedigree blob, and genomic blob. So, and we're gonna also look at different population structures. A classical program, like I said, it could use pedigree blob and blob, but only a genomic prediction-based program could use G-blob as a surrogate of merit. So let me explain these plots step by step. Each of these plots, you can see 3, they have, the only thing where they differ is on the y-axis. So what is the response variable that we are looking at? In some of them, we are looking at the accuracy. In some of them, we are looking at accuracy, just for the information of some. The accuracy is basically the correlation between the estimated and the true breeding or genetic value. So in the case of a classical program, I mean, you can come up with some sort of accuracy, but you actually don't know the true breeding value. In this case, because we are simulating the program, we do know the true breeding value. We know it, so we can actually take the real accuracy, because it's the correlation between what we estimate with any of these methods and the true breeding value. That is, it doesn't require any method, it's the true breeding value. That's because it comes from the simulation. In reality, you cannot get it. Now, let me keep explaining. Within each of these plots, you're gonna see other boxes. In each of the boxes, you can see headers like 10, 20, 30, 40, or 50. What these numbers represent is the number of progeny per family that you arrive at the first stage of yield testing. Let me give you an example. I said we have 1,000 individuals growing in the yield testing. That means that we have 100 crosses, and each cross has 10 individuals. Or you might be evaluating 50 crosses, and each cross has 20 individuals, basically to account for the 1,000 individuals that we are growing. So we were simulating different family structures, or different type of yield trials, because sometimes for some of the programs, it might be that when they go to yield testing, they end up with a lot of families, and each family with very little number of individuals, or the other way around, with very few families with a lot of individuals. So these are different cases. So these are extreme, right? 100 crosses, 10 individuals in each cross, 20 crosses, 50 individuals in each cross. So, and again, these are simulations. So we were able to control all the factors. We also were varying the levels of heritability. So in the x-axis, what you can see are the heritability of the trials. So very low heritabilities, 0.25, intermediate heritabilities, 0.75, 0.75. It was actually going in 0.1, 0.1, so in jumps of 0.1. So basically, we were simulating what happens if a program has different family structures and if the program is growing their trials and they have different heritability. You know, if they have very low heritability for, I don't know, maybe your partners, they don't have a good agronomist or something like that. And we are measuring different response variables. Like I said, the accuracy, but also the response to selection, and also how the genetic variance could be affected by that. And the 3 lines, like I said, represent the methods. So basically, let me explain you some of this. What you can see is what happens under different circumstances. Notice that in the first plot, the higher the accuracy of your yield trials, so the higher the more you move to the right because you have higher, let's say, higher heritability on your field trials, the less effect the G blob, the green line, actually has more accuracy. So, you know, there is less difference between the methods. So, and this is kind of understandable, right? I mean, if you have very high heritability in your trials, really what you're observing has a lot of genetic signal. And then it doesn't matter that much which method you actually use to calculate the breeding value or genetic value for a given trait, because there is so much information in the phenotype that the method doesn't really help that much. But please notice that actually the lower the accuracy in the trial or the heritability in the trial, you can see that there is a massive difference between the methods. Blob is basically assuming that there is no relationship between the individuals. They are fitting a model as random, but even if you have full SIPs, they are assumed to be independent. And this is a typical model used still in plant breeding, right? If you use pedigree blob, this assumes that there is a relationship between individuals. It uses what we saw, the pedigree relationship matrix, and this uses the marker information. So you can see that also something important is that if you actually use very small families, because the G-blob method takes the advantage from borrowing information from relatives, the less relatives you have, of course, you're gonna have less signal and less, let's say, less accuracy, especially at lower heritabilities. You can see that if you have relatively big families, maybe 20, 30, I mean, you can see, of course, at 40 or 50, there is a massive difference at low heritabilities. But if the family is also small, then you're not, let's say, borrowing enough information. So what I can tell you is that it seems that more than 20 is more than enough to actually reach some level to maximize the accuracies. But families of 10, really not that much. Now let me again refresh something because before people get confused. I'm not talking about doing predictions for, you know, reducing cycle time or anything like that. I'm talking about that you go to the, let's say, to the first stage of yield trials. Let's say that is this one. I don't know, maybe you have, like I said, 1,000 entries. You phenotype those 1,000 entries, and you have 3 options. Whether you just fit a model with blob, whether you use the information from their pedigree, so basically the family relationships, or you also genotype those 1,000 individuals and you use G-blob to calculate and you know, some variation you can also use to calculate the breeding value or the total genetic value, depending if it's an inbred or a non-inbred crop. And you can see that basically we are not predicting anything that we have not seen. Basically, we are predicting something that we already see or we already have seen. It's just that we have the phenotypes and we want to put the layer of genotypes on top of that, basically more information to become more accurate. The result, as we mentioned, if the trial was already pretty good, there will be no major difference. If the trial was actually very low heritability, you can expect a big difference. So using the genomic information, actually it becomes some sort of buffer, because even if you have bad trials, you can expect that if you use genomic information, the accuracy of your yield trials will be always high. And that, of course, it gets reflected in the response to selection. So really at the end of the day, it ends up being kind of linear, the response to selection compared to accuracy, but also something that is well known is that normally the use of GBlob tends to decrease the genetic variance quicker, but please don't see this as something negative. It doesn't mean that it's exhausting the genetic variance just faster. It means that it's translating the genetic variance into genetic gain faster. I don't know if there is any question on this plot or any doubt. Otherwise, I'll move to the next example from the simulations that we've been generating in the past 2 years. Okay, so we're talking about just pure accuracy. Like I said, we are not going to talk today about the fancy stuff of using those training models to predict we have never seen, future generations, kind of something that has not been phenotyped. For now, we're just focused on something that we have phenotyped. How can we increase the accuracy of those estimates? Instead of using a mean or a blue or a blob or a pedigree blob, let's put a G blob on top of it. There is an advantage. Big lesson, try to use it, especially if you have low heritability trials, and at least have more than 20 individuals per family when you use those methods. Let's actually move to something that is also quite interesting that one of my members was simulating for all this genotype by environment and TPE, etc. The following simulation is adding another layer of complexity. The experimental design that normally happens in the early generation variety trials, what some people call the stage 1, what other people call preliminary yield trials, and there might be other names for those trials. Here, each box represents a different level of heritability, again, 0.2, 0.5, and 0.8. And the box plots that you can see within each of these boxes represent different experimental designs commonly used in stage 1, when material is normally limited, not that much seed, not that many cuttings, etc., not that many tubers. In the y-axis, again, you can see the accuracy measured as the correlation between the true and estimated breeding value. Again, we can estimate it true because we have simulated these values. In the first box, let's actually, I divided them in these 4, or I separated them in 4, because what you can see here is that in this kind of blue or light blue or gray, I don't know how it reflects in your computer, is basically what happens if you were basically having unreplicated entries, let's say for a minute, those 1,000 entries, but they are not replicated at all. And there is also no checks. What happens if you only make some of those classical analysis? Well, but you cannot do too much, but let's say that you just have the phenotypic record. Maybe you can adjust by, just how is it called? What we did there, the closest, nearest neighbor. Sorry, that's what I was trying to remember. Then in the purple boxes, what you can see is again, a completely unreplicated design. So basically only the 1,000 entries, but what you do to analyze the data is to fit a spatial model. Here you fit a spatial model. Maybe you have seen some of those autoregressive models, AR1 by AR1, using the row and column information that comes from your field trials to adjust the means of the individuals. And in yellows, you can see the augmented designs. You have your entries, but you also put some checks on those trials, and those are called augmented designs. And also some other, let's say now also standard models or experimental designs are the PREFs, the partially replicated designs. So you have your entries, but also instead of replicating some of the checks, if you replicate some materials, the materials that you replicate are also the same entries. So basically some proportion is only appearing once and some proportion of material is showing up, let's say, twice. And what you can get from here is that like I said, these are completely replicated designs, augmented, p-reps, but this happens when you actually add the genomic relationship matrix to the model. At very low heritabilities, as we saw in the previous picture, there is a massive increase in accuracy. This is for a less complex trait. That's why you can see higher accuracies. But you can see that in general, it's the same trend. The higher the heritability, the less of a difference of putting the genomic relationship matrix or the G-block model. But the lower the heritabilities, it has a higher impact putting the genomic relationship matrix in the model. So this is a game changer, especially because many of the field trials in the national and in the CG programs, and I would say almost everywhere, there is a high proportion of trials that don't have high heritability, because especially in the early trials, when replication is low, when material is limited, and sometimes even when the quality of the seed is not that good. So this is really, really important. And like I said, from my perspective, it's a game changer. Something that is relevant for you to know why this is, some sort of animation is because we are changing what is the replication level. Percent checks or P-reps, 10% checks or 10% P-reps, 15% checks or 15% P-reps, 20% checks or 20% P-reps. What you can see as the level of replication increases for the checks or for the P-reps is that in the case of the augmented, actually having more checks doesn't make your model more accurate, by the way. But in the case of the P-reps, if you put more replication, that actually increases it starts to increase the accuracy for those entries. And then the entries start to show up twice. They start to show more, so they become more accurate. But in general, I think the big lessons here is GRM is a game changer. Second, actually that you should stop probably thinking on the checks on something to be used for adjusting the means. The checks are really useful, but they should be used for what you, for what you are actually using them. But just to highlight, to make your visual comparisons on the field, if you want to make some visual comparisons, or also to connect trials to calculate genetic gain. But these days, with the use of spatial models and relationship matrices, actually they're not really indispensable for the adjustment of breeding values or genetic values. Checks are useful, but not necessarily for this purpose of selection. For selection of, complex traits, for selection of visual traits, they might be quite useful. But because many programs tend to replicate a lot the checks, they put those serpentines, et cetera, thinking that those might make them more and more accurate. And actually the gain is marginal from, putting a lot of checks. Normally, like I said, the spatial models and the GRM does a lot of the work when it comes to adjusting or getting adjusted means. When I say here on replicated, augmented, and P-rep, it's just experimental design, just how we put them in the field. So no replication at all, putting some checks and then, partially replicating some of the entries. Then indeed, when I put here LMM, what I mean is the mixed model. And I'm just referring here in this case that we are putting a spatial model. So we are putting AR1 by AR1. So we are using the row and column information from the field to fit a spatial model that basically creates this connectivity between the entries that are in the field and adjust. You can see that there is a jump between not adjusting and adjusting. It might seem a bit marginal, but let's say it's at least from 0.45 to 0.5. And of course, it depends a little bit more on how strong might be the spatial. But that's what I mean. So this one, we are analyzing with a linear mixed model for spatial, this one as well, and this one as well for all of them. And the ones on the top is on top of the linear mixed model, we are putting the genomic relationship matrix, as we mentioned, basically using the G-block model plus the spatial. And you can see that depending on the heritability, as we saw also in the previous figure, at low heritabilities, you expect the major change. It is the same that we observed when we were simulating something for, this was related to GIE stuff. but also to comparing different experimental designs. So again, the game changer is this. So in the case of the actual model, is just an additional line of code. And also here is about taking the genomic information and putting it into the model. That's where basically you need the support from the genotyping, someone that can generate that marker matrix, and your biometrician putting in the model the GRM and the spatial to basically make this jump and be here instead of here. Always my advice is to rely, in your biometricians and your genotyping or molecular breeder support to act as a team and kind of, but it's important, of course, that everyone is aware of how things work. Also, the biometricians, of course, should let you know how the models work, and so you feel comfortable with the results. But you can expect the GRM, like I said, to be a game changer in your program. So let me show you an example on a clonally propagated crop. We're working with jams, and they have as many crops. They have their crossing block, then they produce seedlings, then they have what they call the first clonal generation, second clonal generation. And by the time that they get to the, let's call it the first yield trials or preliminary progeny trials, that's how they call them, actually they end up with very few materials. It's about 60 materials that will be evaluated, but it's because until this point is when they have enough material to replicate in 2 environments with 3 reps. Before, they are only 1 environment with 1 rep. So it's very limited, the material that they have in these generations. So, but because they can genotype very early, and the genotype, in the case of the clonally propagated crops, that is a big advantage, right? They can genotype here, and that genotype is not going to change. Of course, if you don't mess it up, right? If you don't mix the material, but that genotype is not going to change. So in essence, if they are genotyping here, and actually, in the case of the JAM program, what they were doing is that they were actually still, even though they have very limited material here in the second clonal generation, they phenotype for yield, which is one of the most relevant traits as in many others. But because the accuracy comes only from 1 environment, 1 rep, and is particularly small plot, is so small the heritability that they have calculated that they say they don't use that data, and they wait until they are here, and more in general, they keep advancing their products. So what we're saying here is that they could actually use the GBlob to increase the accuracy exactly in the situation that we're mentioning. They could just take advantage from the fact that in low heritability trials, they can gain that big jump and make better decisions for both, for advancing products, but also for recycling materials. So what we did is to take the breeding program and simulate something that what would happen if they were using, again, blobs, pedigree blobs or G blobs, and basically advancing the lines from, like we said, the different generations. I think here is calling them slightly different, but let me just make the point. What we found is that if they were using, by the time that they were making their decision here, now their accuracy that they were reporting, of course, that needs to be checked, but at least they were reporting that their heritability for their trials, it was pretty high. Like they said in these 2 environments, about 0.8 heritability. That's of course super high, but we have to trust what they said. And what we saw, as we mentioned before, is that when the heritability is super high, really the method doesn't matter that much. And that's what the simulation was showing. If you have the phenotype with that accuracy that's so high, and you put the blob and the pedigree blob, I mean, there's no big difference. But because the G blob, actually G blob also would, I didn't show it, I should have put the plot, but there was a two-side coin. There was another plot where basically also G blob was showing up very close, like here. But what made this big jump is when we actually propose, what if you actually use the G blob not in the regular stage that you have already high accuracy. What if you actually put it here when your heritability is so negligible that you almost ignore it? And that's actually when the plot changed from this G blob being here to being here. And this plot, let me explain it because I don't think I did. In the y-axis, this is the mean genetic value, and I don't say breeding value because for the jams, they are a non-inbred crop, so they actually look for the total genetic value. We see the y-axis, the genetic value, and in the x-axis, what we see is the time for breeding. So we simulate that for 20 years, they apply consistently the G-blob method, they apply consistently the blob, pedigree blob, etc. So this is not like the one that I was showing before, where there is different levels of heritability. No, this is the mapping what the breeding program does, exactly their crossing blocks. So this is their breeding scheme, and we are simulating it as they reported with their values, their number of traits, their genetic covariances, correlations, heritabilities. And what comes out, what if you make a change in the breeding program and you start using G-blob to advance? Basically, like I said, the game changer for them was to apply it in the second clonal generation. Then what will happen after 20 years? So because in simulations, we normally simulate 20 years for one program that is running, let's say, a standard. And then suddenly we start to do it differently. We start to do 4 different scenarios. And what is showing is that if they were to start using G-blob, they would find a high increase in genetic gain in the next 20 years. But they will find that not from, like I said, not from predicting new material that they have not seen, just by increasing the accuracy of the phenotypes, of the surrogates of merit, or you can also increase the accuracy of the, let's say, the phenotype somehow, for those trials where the heritability is normally low. So that is something that maybe for the clonally propagated crops, if you are taking measurements for certain phenotypes, but you feel that the heritability is still too low, well, if you have genotypes for those individuals, you might want to start, using those phenotypes that you collected that you think that are not good, and still use them, put the marker data on top of it, calculate G-blob, and you can expect to have a big jump. Not that much in the trials where the heritability is already high. Okay, so we also did another, we were working with the CIMMYT beans program. This is a line crop, and what they were doing, it was to, this is their classical breeding scheme. They were showing us at which stages they were taking parents. So, they were making the crosses, F1, they go through the SSD process, the PYTs, their stage 1, the best 20 materials from those 200 lines that they were testing, they were being recycled. Still, from those 200 lines, the best 60 that were being grown again for a second year, the best 5 were being recycled. From those 60, the best 24 were being advanced, and so on. And their crossing block was around between 35 and 40 parents. So what we wanted to look at here, it was, again, is going to help if we start to use blob, pedigree blob or G blob, because they actually are currently using just blob. This was also quite surprising. Well, at the beginning. Later, we understood why it was such a big change. Here, again, we are looking at genetic gain for the total merit. So this line is representing the selection index for all the traits. There is 8 traits behind this merit. So you can see here the gain for the specific traits, 8 traits. If I remember well, I think it was like 3 simple and 5 complex traits, something like that. And actually, first at the beginning, and I want to ask you to actually ignore this one because this is the current way they do it, just blob, and they don't do any family selection. We knew from a previous simulation that if they were applying family selection, they would make a big gain, because what the beans program does, similar to the jams, is that when they are at this stage of the F2, they do phenotype for yield, but because they are single plants, they of course do not use that information. And also because it's so many individuals, they do not genotype them. But what they were not considering is that they could actually apply some family selection because they were doing 100 process. And even though the heritability for the individual plant is very low, because you have 180 observations per cross, actually the heritability for the cross is not that low. It's actually relatively good, even if like I said, the plant heritability is really, really low. So this is what they found, what we found in some of the simulations is that if they were to use that information to not select the best individuals, but to select the best families, they could make a big jump. But on top of that, and that's why all these three are here, because they are using families selection. But what we wanted to see is going to help if we put GBLOB on top of that. And it turned out that it didn't. But that was because by the time that they were using the GBLOB, it was until they were here in the PYTs, in the first stage of yield trials. And again, we have to believe what they tell us, but what they told us last time when we were doing that simulation is that the heritability that they were achieving, it was 0.54. That was an accuracy of about 0.73 if you take a square root of this heritability. That means that their trials are already quite high in terms of heritability. So the GBLOB is not making a difference because, as we mentioned to you before, GBLOB is going to make a difference when the heritability is low. Like I said, when you want to basically predict individuals that somehow you already observed, which I'll try to emphasize again and make the distinction. So let me just go back to this one. Again, please don't get lost, because in this session one, when we're talking about genomic prediction for accuracy, again, we're not talking about predicting stuff that we have not seen before. We're talking about, we already observed the phenotypes and we just want to make them more accurate by putting the marker data and calculating GBLOB. But again, if this material is already high heritability, you will not gain a lot. But if the heritability is low, you will gain a lot. So this becomes some sort of buffer. And that's why I said it's a game changer, because it can basically ensure that you make genetic gain. More than saying it will increase massively your genetic gain, it will just ensure that you have genetic gain.